I just want to welcome everybody, both remotely and here in person. This is a really an amazing special opportunity to bring in an outstanding speaker. And even though it's still summertime, it's a really good way to kind of get our entree into the new academic year. So I'm going to give a little bit of a formal introduction of our speaker, and then I just give you some of more informal personal reflections. So as most of you know, Dr. Catherine Bushnell is president of the International Association for the Study of Pain. She's also the Scientist Emeritus at the National Center for Complementary and Integrative Health at the US NIH, and also Harold Griffith Professor Emeritus in Anesthesia at McGill. So she holds a Ph.D. in experimental psychology from the American University and received postdoctoral training in neurophysiology at the NIH. She was founding director of the McGill University Allen Edwards Center for Research on Pain from 2003 to 2009, and then director of the NCCIH Intramural Research program from 2012 to 2020. She's received the Lifetime Research Achievement Award from the Canadian Pain Society and the American Pain Society. And her research interests, as most of you know, include forebrain mechanisms of pain processing, psychological modulation of pain, and neural alterations in chronic pain patients. So on a personal note, I just want to mention a few things, especially for the trainees that are here in person and also on Zoom, that if you read through Dr. Bushnell's CV and you listen to her speak, I think what you'll notice is her entire career has been extremely multidisciplinary, and her training coming from different areas has really informed her ability to have great insight into her research findings. So many trainees, kind of wonder, well, what area should I do my PhD in, and where will that take me in the future, and what should I learn in terms of technology and subject matter, because who knows where the future is going to be. So I think this is a really very instructive to see how wonderful it is to start in one area that gives you kind of a pathway to the future. So maybe you've already spoken to the students about this already, but I know you did work in your doctoral years in the visual system, right? And in psychology, you became an electrophysiologist working on different models of pain and how task-related responses in various areas of the brain related to pain. But also then all of a sudden, PET imaging, the PET technology was developed, and Dr. Bushnell was the first to take the opportunity to apply that to pain studies. So to be an imager these days, a lot of people are training as imagers, but I think it's really important to have a good foundation in some of these other areas, which is really instrumental to being able to interpret your data, and also to have a good foundation in behavior and psychophysics, because to do these brain behavior relationship studies, you need really the insight from all those areas. So I think in that way, she's really been a role model in how to navigate a career in a changing landscape of technologies. And of course, when what I was seeing when I was developing my own career is also a role model in terms of women in science and neuroscience, and really paved the way for many of us throughout the years. So it's really my pleasure to have you here in Toronto, and her talk is entitled Beyond Opioids, Engaging Endogenous Pain Models in the Brain.
I am retired and I basically, when the pandemic hit, that was the last time I was in a lab and collecting data. So I don't have any new exciting, data right off of the press. So this is kind of a retrospective on things that I've done and my lab has done over a number of decades. So I will not be talking about hot new technologies, but I'll just and I'm sure many of you, Karen's lab was working kind of in parallel with our lab, doing a lot of the same things. So many of you have done things very, very similar to what we did. But So, you're more familiar with this, but hopefully, it's not going to be all a repeat of everything you already know. But okay. So, and I was also didn't expect to have, so I was told there was going to be hardly anybody in the room. It's mainly a Zoom con, but there's a lot of people here. So considering we're going into a holiday weekend, so thank you all for coming. Okay. So disclosures. Now I don't do anything, so I don't have to disclose anything. So the opioid crisis has raised questions of how can we treat pain without drugs? And much of my work over the years has focused on how drugs work in the brain to reduce pain and how we can engage these systems without drugs. So today I will talk about how pain is processed in the brain, how systems in the brain work to dampen or augment pain, how chronic pain can change the brain, some lifestyle choices that can help reduce pain. So again, many of you already know all of these things. We published this, 10 years ago, based upon work that Karen and we and many others did of putting together the pathways of pain in the human brain. And what we all discovered is that there are multiple parts of the brain that are involved in processing pain in humans. And there's the multiple pathways correspond to the complexity of the pain experience. So, we all know we have the pain sensation, the intensity, the quality, the pricking, burning, aching aspects of pain. But we also have the emotional aspects, the unpleasantness and the suffering. And as you're all well aware, it is that suffering that actually drives people to go see their doctors. You don't go to the doctor because, you prick your finger with a needle and it hurts. And so the suffering is really what people talk about when they talk about having chronic pain. So in the cerebral cortex, pain sensation and pain emotion are processed generally differently. I'm giving you kind of the almost like the layman's view of this. I know many of you who are really into this know that it's more complex, but in general, using MRI, as Karen and we have done, we can look at how pain sensations and pain unpleasantness correlate with activations in different parts of the brain. And we find that in general, pain sensation is primarily processed in the somatosensory cortices, S1 and S2, whereas pain emotions involve regions such as the anterior cingulate cortex, the insular cortex. Again, this is not exclusive, but it's a kind of a general way of looking at it. So we all know that people react very differently to the same painful stimulus. Remember when my daughter was growing up, she's like the girl on the right. Anytime there was going to be a needle coming, she was like underneath the chair, hiding. Whereas other people, it's no big deal, right? So what accounts for these individual differences in pain perception? Well, there are also multiple descending pathways in the brain that can dampen or enhance pain information. Just as there's multiple afferent pathways, there's multiple descending pathways that can alter the pain experience. And some of them involve like the prefrontal frontal cortex, and others involve like the posterior parietal and descending pathways. And different people may have different abilities to engage these systems in the brain. So there's high opiate binding throughout all these descending pain modulatory systems. So can't see my slides very well. In the thalamus and in the, sorry, this is like too small for me to see. Anyway, multiple areas, anterior cingulate cortex, the putamen, the caudate nucleus, the amygdala, regions that we know are important for pain modulation. There is higher opioid binding in those areas than some other areas of the brain, even though there's opioid receptors throughout many, many parts of the brain. So are these opiate receptors related to pain and to pain modulation, or are they related to other aspects of the opioid experience, the euphoria, other aspects of pain, of that, other things that happen when you take an opioid other than reducing pain? So this can be studied using positron emission tomography, and this has been used by multiple labs, but and this is what I'm talking about now. It was not from our lab, but mainly from the work of Jean-Claude Zubieta and Rick Harris. where they used carfentanil, which is, I guess everybody knows carfentanil now. People didn't used to know carfentanil until the opioid crisis, but it's a very, very potent opioid agonist, and it's used for PET, given in very microscopic doses in order to look at the endogenous release of opioids. So you inject the ligand, and it's going to be taken up in the brain. But if the receptors are already occupied in the brain, then they can't be taken up at those spots. So if you give a pain challenge, so that if the pain is actually leading to the release of the opioids in a certain area, then you give the exogenous opioid that's radio labeled, is not going to be taken up in these areas where they're already bound. So you can look at the difference in the opioid binding from this radio labeled exogenous opioid when the person is in pain or not in pain, to see where pain may be leading to endogenous release of opioids. So when you do this, and this is some of the classic work of Jean-Claude Zubieta, where they used a masseter hypertonic saline injection in the masseter muscle and created facial pain, and they compared that to no pain. They found that it was reduced opioid binding in a number of regions of the brain, suggesting that the pain had caused the opioids to be released in these areas. And then they looked at the various areas where there was this change in opioid binding, and they looked at the correlations between the person's pain experience using McGill Pain Questionnaire. And if you're well aware of the McGill Pain Questionnaire, there are questions that are related to the sensory experience and others that are related to the emotional experience. And there are certain regions where the change in activation was related to the sensory scores, and there were others where they were more related to the affective scores, like particularly in the anterior cingulate cortex and the contralateral thalamus. But overall, there were these number of areas where clearly the that pain seemed to release opioids, and it was related to the pain experience about whether or not the opioids were released. So how does chronic pain affect this endogenous pain regulation? So descending pain modulatory circuits, there's more and more evidence, and this is accumulated over the last 20 years now, where that circuits are disrupted in chronic pain patients. And I know some of you that I talked to today are actually studying this now. So one thing that's been shown by many, many studies now is that chronic pain patients have reductions in brain gray matter, particularly in regions involved in pain modulation. So this is a study done by Dave Seminowitz. Is Dave at UT now? Where is he? Western, okay. So, and he worked with Karen, and then he worked in our group. And this is a nice study showing that chronic back pain patients have less gray matter in pain modulation regions of the brain. So now the color blobs that you're seeing now are not activations, and they're not opioid release. What they are is regions where there's less gray matter in the pain patients than in matched healthy controls. So as those of you who are not doing human brain imaging, you see all these colored blobs, and they can mean whatever. They're basically a way of graphically showing the statistical differences. So it can be activation, it can be gray matter, it can be opioid release, it can be whatever, but it's just showing the differences. So here you see that there are large regions, particularly in the prefrontal cortex, of reduced gray matter in the back pain patients compared to the healthy controls. And Dave, along with Marta Checo and others in our lab, showed similar findings with fibromyalgia patients, again, in these areas, dorsolateral prefrontal cortex, anterior cingulate, medial prefrontal cortex, similar regions with fibromyalgia. And now there are studies in many, many different types of chronic pain. Each study might have slight differences in the regions, but overall, you get a lot of these changes, particularly kind of the regions, areas that we know are important for pain modulation. So are these gray matter reductions caused by pain? These are correlational studies, right? So we don't really know. We can't show cause and effect. And it might be that, say, people that just don't have very good dorsolateral prefrontal cortices, are more susceptible after to come down with chronic pain conditions, or to have for a pain chronic after an injury. So we can't tell this from these correlational studies. So one thing that we've done over the years in our lab is we back translate findings from human studies back into animal models, which I know Karen also does this. And this, again, this was work that Dave did in the lab, where he took mice, and half the mice had an SNI injury, a nerve injury that led to chronic pain condition, and half the mice have a sham control, so they were genetically the same, everything environmentally the same, but then we followed them over time after the injury and looked at, with a little MRI scanner, looked at their brains and found that by 15 weeks post-surgery, the rats were continuing to grow, so the sham treated animals, their bodies got bigger and their brains got bigger, which is the blue line going up as they grew. And you can see that the ones that were injured, their brains didn't grow at the same rate. And by 15 weeks, there was differences, particularly in the prefrontal cortex, in the amount of gray matter in the injured animals versus the healthy animals, suggesting that something about the injury or the pain led to the change in gray matter versus the fact that they were different, because these animals were basically genetically the same before the injury. Now, there's also evidence that chronic pain alters opioid receptors in the brain. So again, you go back to the PET with carfentanil, and Rick Harris and his group compared patients that had fibromyalgia with those that were healthy, and they looked at the opioid receptor binding and found that there was reduced binding of the exogenously administered opioid in fibromyalgia patients compared to healthy people. And the areas that were significant were ACC, amygdala, nucleus accumbens. And again, there's been other studies. Again, there's little differences in the exact areas that become significant in the different studies, but in general, there's been similar findings in multiple pain conditions, including arthritis, neuropathic pain, headache, complex regional pain syndrome, suggesting again that chronic pain can lead to reduced opioid binding in the brain. But again, cause and effect, we don't know that. It might be that people with chronic pain who just don't have a great opioid system. So what does it mean that we see these things in these studies, these correlational studies? So could it be related to the opioids that the people with many people with chronic pain have taken opioids? If they don't take them at the moment that you're doing the exam, they have a history of taking opioids. Or is there an endogenous release of opioids in response to their chronic pain? If they're in chronic pain, then that could lead to the endogenous release. Is that the reason why the receptors are not available? Or is there been a change in their brain due to the chronic pain so that they don't have as many opioid receptors that are available? So again, we went back to a rodent model in which we could control the environmental factors, including their drug use, and examine possible brain changes at the cellular level. So the questions we asked are, do we observe using PET reduced opioid receptor binding potential in a rat model of chronic pain in opioid naive animals? And what is the cellular molecular basis of changes in the opioid receptor availability? Are there fewer neurons? Again, with the human models, reduced gray matter, is that related to fewer neurons? Could there be, again, increased endogenous release because of chronic pain? Or could there be a decreased receptor expression? So again, we went to use the SNI, the spared nerve injury model of chronic pain. And now we did PET scanning in our rat model, and we didn't have carfentanil. We used diprenorphin, which is it is not as selective for mu. It's less selective, but it's opioid receptor antagonist, not an agonist, but it's less selective for the mu receptor as carfentanil. So again, we did this over time. We did our pain testing, but we also did sucrose preference testing to look at possible anhedonia related to opioid that we see with opioid use. So we did sucrose preference test, we did sensory testing, then we let them go for a while. And then at week 11, we did the test again. Week 13, we did the MRI scan. They don't actually lie in a scanner on their back, but you know, it's okay. So 3 weeks post-injury, we had the same, basically what everybody finds with SNI. You have this protracted change in mechanical sensitivity. They're hypersensitive to touching their foot, and they are hypersensitive to cold. But we also found that there was difference in the SNI and the sham animals in terms of the sucrose preference, while 3 months after the original baseline, the sham-treated animals actually had a higher preference than they had before, whereas the injured animals did not. So there was a significant difference in their preference for the sugar water at 3 months. So again, we looked at just the binding in the sham-treated animals to see if it was similar to what you see in humans. And again, as with humans, you see high binding in areas like the amygdala, the PAG-ACC, nucleus accumbens, prefrontal cortex, thalamus, same regions as you see high binding in the human studies. And then we looked again at the binding, the difference in the binding between the injured animals and the healthy animals. And just as Harris shows, you know, with the fibromyalgia patients, we found reduced binding of the exogenous opioid in multiple areas, with the main area, the most important areas being the insular cortex and the caudate putamen area. So knowing that, then we went back and we took the tissue from these areas where we found the reduced opioid receptor binding, and this is the royal we because I didn't do this, the people in the lab, used immunohistochemistry to look at these various areas. So here, to look at possible reduced gray matter reduced numbers of cells, we used NuN immunoreactivity, and to look at possible increased endogenous opioid release, we used enkephalin binding, and then for decreased receptor expression, we used MOR1 immunohistochemistry. So when we did that, we found that, in fact, when we counted cells using NuN, there was no difference in, so we did this in the anterior insula, the posterior insula, and the caudate putamen. And we found no differences in the number of neurons in these regions. We found no differences in enkephalin staining, but we did find significant differences in the MOR1 receptors and the receptor availability of the animals that had the injury versus those that didn't. So we concluded that the reduced receptor availability observed with PET is related to fewer mu opioid receptors. So that's really a change in the brain, and not just the fact that there are drugs on board with people, you know, if we take this back to the human. We also found that the reduced receptor availability, particularly in the caudate putamen, correlated with this reduced sucrose preference in the injured animals, both correlating with the PET data and with the MOR1 data. We found that, again, the more reduction there was, the greater less the sucrose preference there was. So these results suggest that pain-related anhedonia may at least partially be mediated by reduced opioid receptor availability in the brain. So given that pain modulatory systems may not be working as well in chronic pain patients as in healthy people, it is particularly important to find ways to engage these systems without resorting to opioids. So as we all know, analgesic drugs, including opioids, work through activation of pain modulatory systems in the brain. I mean, there's some peripheral activation as well, but most of your analgesia comes from centrally acting drugs comes from the brain, so that you find opioid receptors throughout, from the cortex on down to the spinal cord. But there's, also other drugs that we find that are analgesics also are working in the brain. So, but can we activate these whole descending control systems without using drugs? And this is something that I've studied over decades. Karen has studied over decades, and we may be the ones that have studied it more than anybody else, although it's becoming more in vogue now. But we know that emotions can alter pain. We know that attentional state, attention versus distraction, can alter pain. And we also have learned that attention emotions actually change different aspects of pain. So that when you have subjects rate how intense the pain is and how unpleasant the pain is, and you do attentional manipulation studies in which you are, have them focusing on the pain or focusing on something else, at least in our hands, we found that the pain intensity, changes in pain intensity scores change more reliably than changes in pain unpleasantness scores. But conversely, when we do manipulations that change the emotional state of the person, putting them in a positive state or a negative state, we find that there is a preferential change in their perceived unpleasantness without actually changing the perceived intensity of the pain. They still feel the pain, but they find it less unpleasant when they have the pain and they're in a good mood versus a bad mood. So again, this is being a little simplistic because there's a lot more, but in general, pain attention modulates sensory pathways. And this is an old study where looking at primary somatosensory cortex, when a person is focusing on the pain, that's on the top, the brighter spot, meaning more activation. Then when the person is distracted and paying attention to something else, you get less activation. The primary somatosensory cortex, when the person is distracted and when they're focusing on the pain. Whereas mood alters emotional pathways. And again, here's the anterior cingulate or mid anterior cingulate cortex, and you give the person the pain and they're in a bad mood, you get a nice robust activation. You put them in a good mood, you give them the exact same pain stimulus, and this time it doesn't even reach statistical significance in this particular study, suggesting that you're getting more activation in the cingulate cortex when you have pain in a bad mood than pain in a good mood. So the attention emotions activate different modulatory circuits. If you take and correlate these changes with these various pathways, what we found is that this pathway from the superior parietal lobe down, through the insula and down, going down, that pathway is more activated with the attentional modulation, whereas the emotional pathways involving the prefrontal cortex are more activated when you by the emotional modulation. And interestingly, this superior parietal lobe, this pathway is important for visual attention as well. So attention affects all our sensory modalities, and it's been studied a lot in the visual system. And the same superior parietal lobe is very important. It's been identified as being very important in visual attention. So I think this attentional circuitry in the brain can affect all our senses, but in pain, it leads to analgesia. Like, when with the visual system, you can be, hopefully you're paying attention to me because I'm speaking, but you can be looking away and you can still be paying attention to an object elsewhere. And there's been a lot of work about that whole system, about how attention, because you see everything, you've got this big visual field. So why do certain things come out as being more important than others? And again, this same area is very important for visual attention as we find for pain. So can lifestyle choices engage descending pain control systems without drugs and reduce the impact of chronic pain in the brain? So we've studied some of this as well throughout the years. So one thing is yoga, exercise, various types of active lifestyle. So how about yoga? Lots of people are using yoga now for pain. And there's actually now been a meta-analysis, it's hard to see the details, but if you see that most of these lines, most of the means are over to the left, meaning that this is showing their pain and chronic pain in people that low back chronic chronic low back pain in people who are when they are practicing yoga, and you can see after they go through start practicing yoga, both the short term, then medium term, and then long term, there seems to be less chronic pain when they're practicing yoga than not. So there hasn't been a whole lot of quantitative studies on this, but it's starting to come about, and you start seeing these meta-analysis suggesting that practicing yoga can be very useful for chronic pain. So we've taken this into the lab and compared pain perception in yoga practitioners versus healthy controls using the cold water tolerance test. And what we find is that the pain tolerance to cold water was much higher in the people that practiced yoga. We just took people who had practiced yoga for at least 6 years, at least a couple of times a week. They weren't like crazy yogis, but people who practiced it regularly, and they had a much higher pain tolerance than those the control subjects. And then when we looked at these, we did brain imaging on the yoga practitioners versus the healthy controls. What we found is that doing anatomical imaging, that the yoga practitioners actually had more gray matter than controls in multiple parts of the brain, including regions involved in pain modulation. So whereas chronic pain patients show all these regions with reduced gray matter, we found that compared to matched healthy controls, the people that have been practicing yoga actually had like widespread areas of increased gray matter. And if you look at the relationship between gray matter and age, unfortunately for, I'm actually off the chart here, unfortunately, but that your gray matter decreases with age, and that's a very robust phenomenon. But the yoga practitioners, here are the chronic pain patients, you see this accelerated gray matter loss, and Karen has shown this as well. But that with the yoga practitioners, there's actually a decelerated gray matter loss in the yoga practitioners compared to healthy controls. And then we looked at this gray matter, the relationship between gray matter and this pain tolerance difference that we found with the yoga practitioners. And we found that in one area, the insular cortex, the gray matter size actually correlated with pain tolerance, suggesting that there may be a relationship between this gray matter changes and pain tolerance. Again, it's only correlational. So now we're going back to cause and effect. Gray matter is just, is it that people who practice yoga just have better brains to start with, or is it practicing yoga? Does that actually lead to this change in gray matter? So at this point, we were not able to go. We weren't willing. Maybe you could do it. We weren't willing to take rats and have them do yoga. Hélène Langevin, who is the director of NCCIH, she does a lot of work with stretch. She basically does yoga with rats, which is pretty amazing stuff, but we didn't do it. So we took another approach, which is not as robust, but at least it gives you some indication. You can look at, are the gray matter increases in the yoga related to the length of the yoga practice? We had people that had practiced yoga for 6 years and others for 25 years. And so you can guess that if there's, the longer you practice yoga, the more gray matter you have. It's a suggestion that there may be a causal effect. It doesn't prove it, but we did find a very robust phenomenon that the longer the people have been practicing yoga, the more gray matter they had in multiple parts of the brain. So we also, because we took people that did all these different kinds of yoga, we found that there was a lot of differences in the way they practiced yoga. So then we asked them, like, how much of your yoga practice did you devote to the postures, the exercise part? How important was that to your yoga practice? And then how important was breath control, and how important was meditation? There's certain people that all they wanted to do is do the exercise, and they didn't worry about their breath. They didn't meditate, and others that were, and you can see there's a lot of differences in the way people attributed these different aspects of yoga. So we took this variability and we used it as a way to look at the importance of the different aspects of yoga and how that contributed to the gray matter increases. And in most of the regions, we found that the postures, the exercise part, had the largest contribution, although there were some areas, oops. What happened here? Okay. Some areas where the meditation was the most important contribution. But again, overall, the most important contributor to the effects of the yoga on the brain seemed to be the exercise component. So there's growing evidence that people who exercise have reductions in chronic pain. These books, you go to the airport and you see all these books about, about exercise and pain, but there now actually is more than just this kind of anecdotal stuff going on. There have been a number, this is really, you can't read this, but you see that these are a whole lot of studies that have looked at chronic back pain and gray matter volume, chronic back pain and exercise. And you can see, again, this line down the middle, anything to the left means that people who were exercising had less chronic back pain than the ones that didn't. That's a lot of studies. So even though they're not all wonderful studies, when you get that many studies showing that people that exercise have less chronic back pain than those that don't, I would say exercise is important. And there's been a little bit of work of imaging work to show that here, actually, they gave an exercise program to healthy people, and they found that even the healthy people, just like with the yoga practitioners, there was an increase in gray matter in multiple regions when the person exercised. So can exercise reduce chronic pain in anyone? Or are people who are willing and able to exercise essentially different from those who don't? Because everybody knows you're supposed to exercise, right? Everybody, but how many of you exercise regularly? And here we're talking about young, intelligent, healthy people, and you still don't exercise. So people, they go to their doctor, they got back pain, say you should exercise. Okay, sure, but they don't. So again, in order to say, is it that some people that are willing to exercise, they're just have different brains or different that? So again, we go back to our rats and we say, okay, rats are all the same, and can we be tested using these rodent models? So why am I getting stuck here? What's that? Okay. So here first of all, there are a lot of studies of forced exercise in rats, and there are studies in mice where you put a running wheel in their cage. If you put a running wheel in their cage of a mouse, they're like compulsive. They'll run like 15 kilometers and they're like, this big. So it's crazy. It's not anything that you would actually do that translates to humans. And if you then take a rat and you put them on a treadmill, and if they stop exercising, they step off onto electric grid, that might actually be something that you could use in a gym that would motivate people. But since we don't, it's not the same. And that obviously that causes stress. And we all know about stress-produced analgesia. So there's all these studies about exercise and pain, but they're not really relevant in my mind. So we took rats and we sent them to the gym 1 hour a day, 5 days a week. So that's not an unreasonable thing. So they went from their home cage to a cage that had a running wheel, and the control ones they also went to the cage, but it was locked and they couldn't actually run in it. And then we used a CFA arthritis model, inflammatory pain, which we felt was very relevant to many people who should exercise. And first of all, we said, okay, well, rats run if they're in pain. Maybe they just won't run at all, because we've also observed that you put a running wheel on rats, just like people who get that, their exercise bike and they use it as a clothes rack in their bedroom, that rats can also just, oh, this is a nice place to sleep. So the first thing we wanted to know, do the ones with the injury, with this arthritic injury, do they run as much as the rats without the injury? And you can see here, there's a huge variation. Some rats like to run, other rats don't run at all. But there's no difference between the sham and the CFA animals, but you see there's a huge variation across all the animals. So is this normal, moderate, voluntary exercise analgesic? Well, we actually found it was incredibly analgesic to our surprise. First of all, we did a weight-bearing, again, very relevant model. Do they limp or not? And we found that the animals that were sedentary and had the injury, they limped a lot week 1, and they continued to limp week 2 and week 3. You see the ones that were allowed to run, even though they limped initially, by week 3, they were not limping at all, even though their joint was still highly inflamed, highly swollen, but they were back to normal behavior. And then we did this foot withdrawal to heat. And in fact, the ones that were going to the gym, the sedentary ones were hypersensitive to heat throughout the whole 3 weeks, while the ones that went to the gym never showed this hypersensitivity to heat. So then we measured corticosterone, the cortisol, to see for stress response. And we found, okay, baseline, they were all the same. 3 weeks later, the ones that were sedentary and had the injury had increased stress response, whereas after 3 weeks of conditioning, both the sham animals and then those that had the injury and that were allowed to run basically had reduced stress, and it was basically stress to the same as the sham animals. So it was a big difference in stress response if they were allowed to run or if they were sedentary. Again, they all went out of their cages into this other room, but one was allowed to run. So similar to yoga in humans, voluntary running in rats increases gray matter in the brain. And we didn't do this, but just to show that, in fact, you can see this in animal models, that there is a change in gray matter in animals that are running. So what is all this? I know I've gone through a lot of stuff and a lot, 30 years of research, but we have these systems in the brain that modulate pain, and opioids and other drugs activate these systems. We know that. Oh no, not again. There we go. Okay, got it. Okay. Okay. So I hope I've shown today that we can activate these same systems through psychological state, attentional state, emotional state, and that an active lifestyle that involves both these positive effects on attention and emotions, as well as exercise, can reduce chronic pain by accessing these systems and normalizing the brain. So I just want to thank, there are many people that have in my lab over the years, and these are the people that did most of the work that I've talked about today. And then I also just want to make a plug, ISP, those who are not members of ISP, please think about joining it. I know it's hard. You've both CPI, you're already in Canadian Pain Society and maybe other societies. But again, as we were talking today, going to the ISP meetings, and if you're going to go to the meeting, you might as well be a member. It's cheaper to be a member than not to be a member. And accessing our pain research forum and our early career network and all these other aspects, it's worth your $50 to join it. And here we go. I figured it out. So I was talking today about pain research forum, which I think it's the only way that I keep up with the literature these days. I don't know about others, but it's just like it does it for you. You go look every week, papers of the week, and it really helps because there's so much out there. It's not, it's in so many different journals. And I hope to see everybody in Amsterdam, please, and all you advisors. Make sure your trainees go. And we're talking today how great it is to get back to meetings. People are going to NUPSIG, and getting back, seeing people, being able to talk about your science and that your ideas around with others. It's invaluable. So thank you all.